November 16, and you're here, there, and everywhere for your weekly dose of brain science with Pospo and BRAINCAST for mostly learning. Last week, we delved into the abyss of neurological and neuropsychiatric complications of COVID-19. And I say abyss as things, unfortunately, are not that clear at the moment, with considerable more research needed. And thank goodness for Dr. Tim Nicholson and the rest of the COVID CNS study who will be doing exactly this. You missed last week's session? Don't you worry about a thing. All our episodes, starting from the very first one back in April with Dr. Tom Pollack, they're all free for you to access, both as videos on YouTube and podcasts on Spotify or whatever else you're using. This week, though, we delved deep into our brains. No, not the basal ganglia bit. I'm talking proper deep, cellular level deep. With me, a scientific Medusa. And I say Medusa because he has had so many heads. Former head of the Cells and Behavior Unit, head of the Division of Advanced Therapies at the UK National Institute for Biological Standards and Control, and director of the UK Stem Cell Bank, with studies at UCL and MIT, but also leading positions in the industry, like research director at SmithKline Beecham. He is a professor of developmental neurobiology with specific interest in the commercial and clinical translation of gene and cell therapies. And to be honest, who better than to talk to us about the future? BRAINCAST people, this is Professor Jack Price. Prof, welcome to BRAINCAST.
Thank you very much, Pospo, and thank you very much for the invitation. It's a pleasure.
So, Prof, let's start with the basics. What is a stem cell?
Yes, well, what's a stem cell? Well, it's a very general term. Basically, it means a cell, a primitive cell that has the capacity to give rise to other cells. And so, you know, they dominate in a number of different tissues that we know about, the blood being the most obvious. So they're primitive cells that can generate lots of other things.
Wow, okay. And how early do I have to go back in time to find the first mention of stem cells?
Well, the term was pretty common currency amongst 19th century embryologists, particularly German embryologists. Ernst Haeckel is credited with the first use of the term eine Stammzelle. But it was common amongst, as I say, German, particularly embryologists at that time. In more modern times and in a more biomedical context, the story really starts in the 1960s with James Till and Ernest McCulloch, two Canadian scientists who really discovered the bone marrow stem cell, the blood stem cell. And that really formed our thinking about stem cells really for the next generation. A lot of what we learned from those cells, we still apply more generally now. And of course, they're still therapeutically the most important stem cell population. I mean, our hematological colleagues use them all the time for the treatment of blood cancers and so on and so forth. In the nervous system, the story really starts about the same time, but it's got a much more checkered history. There was a guy called Joseph Altman who really discovered stem cells in the hippocampus, again in the 1960s, but he was widely dismissed and disbelieved, and it was another couple of decades before it really got taken up. And of course, it's since been carried forward. We now know it plays an important role in depression and lots of other human conditions. Our colleague Sandrine Charette here at the Institute carrying forward a lot of that work. So there's a lot of very important stuff in the brain now we know about.
Now, for many years, Prof, we took for granted that mature cells are just that. So cells destined to spend an eternity being what they are. Essentially, a skin cell was trapped being a skin cell and a blood cell being a blood cell. But in 2006, Shinya Yamanaka freed cells from the destiny. So can you tell me a bit more?
No, it's a very good way of thinking about it. It freed cells from their destiny, and it freed scientists as well. I mean, it's very common now to talk about breakthrough technologies, but that really was. As you say, he worked out a way of taking basically any cell, skin cells, blood cells. In our lab, we would pluck scalp hairs, and from the few little keratinocytes you could grow off the root, we'd use those. That worked fine. I've got other colleagues who spin the scuffed up bladder cells out of a urine sample and use those. So, I mean, you really can use anything. And exactly as you say, you can then reprogram them to make what we call pluripotent stem cells. So pluripotent meaning really they can make anything, any cell type in the body. And you end up with basically an immortal line of cells that you can then turn into anything you like. So it really was groundbreaking and totally transformed the way we think about stem cells. Yeah.
Now, one of the main difficulties that researchers all around the world face, when studying psychiatric disorders, is that the brain is, let's just say, not that open to others, tucked away inside our skull. It doesn't really allow us to see, what's happening up there. But these techniques of the iPS cells actually changes that, as you could in fact create neurons from skin or blood, or as you just said, anything, right?
Absolutely. Yeah, I mean, I always feel sorry for our psychiatry colleagues. I mean, if you're a child psychiatrist, for example, and some child comes in with the mother and you do a few tests, you talk to the mum, you so on and so forth, you come to a diagnosis, say, of autism. And I kind of feel sorry because you're there faced with the fact that it's kind of everything that's really important has already happened. Everything you really wanted to know about took place in utero. And you come along and it's much too late, really, I mean, there are therapies, there are things you can do. Of course, it's not hopeless. I'm not suggesting that. But everything of importance has already happened. So exactly as you say, what these iPS cells allow you to do is to grow these pluripotent cells as brain cells in a culture dish and follow them through that otherwise invisible period of fetal development. When they develop as nerve cells, they make synapses, they make contacts, they make structure. So you can see all of those things now unfold in a dish using human cells. You're not having to resort to a mouse or a rat or a rabbit. These are human cells behaving for you in a dish. I think it really is a breakthrough technology.
I mean, it's funny because, all this at first may sound so removed, from clinical reality, but essentially they are, the pinnacle, the absolute essence of what we call, personalized medicine, right? So can you give us an example of, how iPS cells can progress our understanding of psychiatric disorders and subsequently, hopefully at some point, influence our clinical realities?
Well, I think I would say there are three areas where it's going to really make an impact. The main one at the moment is that it allows us to model disease. So, for example, a lot of psychiatric disorders have genetic risk factors, don't they? So we can take cells from a patient that's got a particular genetic variant associated with autism, schizophrenia, whatever, and we can grow those cells in culture, and we can simply ask, how does the development of those cells differ from these cells that we've taken from neurotypical patients? And so we can start to model a disease and see exactly how early things are happening. And one of the things we've learned is that things happen much more early than we'd previously perhaps guessed. We'll probably talk about that later, I would imagine. So that's one thing, we model disease. There's also assay development because, we can test drugs, we can develop biomarkers using those cells. I mean, let me give you an example. So some colleagues at Harvard used cells derived from patients with motor neuron disease, with ALS, and showed that they had this hyperexcitability. They could then look for drugs that modulated that, and they came up with a drug that already existed, retigabine, which many of our colleagues will recognize as being a drug that was used for epilepsy, for fits. And that drug is now in clinical trials for ALS, right? So that's a real development that grew out of the motor neuron. And then iPS cells coupled with transcriptomics is revealing lots of new potential drug targets, druggable targets that are different in in autistic, schizophrenia, depression, whatever, those iPS lines compared to control. So that's another area that people are following up.
Amazing. I mean, as a scientist, you have a specific interest in autism. And it was only in August that you published in Science Advances a paper using iPS cell-derived neurons. So let's start with the practical aspects. So how long does it take and how much does it cost?
Well, I mean, IPS work is expensive work. There is no question. that said, like most of these things, it's getting sort of cheaper, and it's now, I think it's at the sort of expensive end of tissue culture work, but it's within the normal, price range that tissue culture studies normally do. iPS cells have expensive tastes in culture medium, but that apart, they're sort of, you can manage them. It takes probably about a year to make an IPS line, but look, here's the thing now. IPS lines are being shared worldwide. I was part of a project finished a couple of years ago called STEM Bank, where we made 500 IPS lines and shared them. Right, so you don't need to make your own lines now quite frequently. There are lines that can be shared, and people, there are banks of lines that have been established. You mentioned that I used to be director of the UK Stem Cell Bank. We've got almost 200 pluripotent human lines that people can just gain access to. So, so cost actually in making the lines isn't really rate limiting at the moment. And actually, it's worth perhaps mentioning, Pospo, that has increased dramatically the power of the kind of studies that could be undertaken. I mean, a recent publication from Lorenz Studer's lab, for example, they used 30 isogenic lines, right? 30 lines that were all genetically identical, except each carried one different mutation in an autism risk gene. And they could compare, and it allows them to find common pathways and common mechanisms. They're really important. So really powered work now.
Amazing. And what did you find in your paper, the one that we're just talking in the autism one, in the Science Advances?
Well, it's very kind of you to call it my paper. I mean, Kate Ware Cornish and Leo Perfect did all the work. And I should also say that you mentioned that I worked for the MHRA for a period of time. I sort of ran out on this project 2/3 of the way through. And if it wasn't for Deepak Shrivastava, my colleague who picked the work up, it would never have got finished. So I'm very grateful to him. But what did we find? I mean, it's one of a number of studies that have come through now that are really starting to indicate that some of these autism risk genes have an effect much, much earlier than people had previously thought. So, for example, a lot of the genes that we know about are associated with synaptogenesis. We know that from lots of old biochemistry and cell biology. But in fact, a lot of them have effects much earlier in the life of a neuron. And we were able to detect impacts really right at the moment when neurons become postmitotic. So really what would be equivalent to sort of 12 weeks in utero, very much in the first trimester. So that's the sort of general thing. What we found specifically was that we already knew that autism, a risk factor for autism was maternal immune activation. In other words, if mum gets a serious viral illness during her first trimester, then her risk of having a child with autism, subsequently diagnosed with autism, goes up really quite dramatically. And what we were able to show was that various cytokines, the one we mostly worked on, one called interferon gamma, seems to interfere with, so that's a cytokine produced by the mum in her neuroinflammatory condition. Yep. It crosses the placenta, impacts fetal development. And what it does is it changes the shape and the mode in which the neuron develops. And the interesting thing is, it works via molecules that we've known about for years, the MHC class 1 genes. And it's a perfect example of convergence of evolution. So we know about MHC being important in immune activation, in self versus non-self recognition, all of those things. But perhaps less well known is that it also modulates the behavior of neurons, how they grow, the connections they make. And so by activating these MHC genes in baby neurons, fetal neurons, you're affecting their wiring. And we think that's what goes on to have an impact on, in autism downstream.
Amazing.
And we've also found, I mean, I don't want to go into a lot of detail, but we think we've identified the memory mechanism as well, because this change in MHC is happening at the precursor cell level. It's before the neurons are even born. But the neurons subsequently downstream remember they've had this earlier insult, and it changes their behavior downstream. So and we've discovered what we think is part of the molecular mechanism that drives that as well.
Amazing. I mean, now, although iPS cells, may be a great tool to study, as you've just described, the neurodevelopmental disorders, given that, neurons differentiated from iPS cells most closely resemble cells of the fetal brain. So isn't that a problem for modeling diseases that emerge later in life?
Well, yes. I mean, if you really need sort of aged nerve cells that you might really like for, say, neurodegeneration studies, then it's a problem. But there, I think I would say you've got to compare with what we've already got. I mean, the systems we've already got don't have that aged property. I mean, most studies of for example, transgenic mice use young mice. They might carry the APE variant, ApoE variant associated with with Alzheimer's, but they're still young mice. So it's a generic problem. But I would say that there's sort of two things. The one is that the genes help, genetics help. So, for example, in Chris Shaw's lab, they're looking at motor neuron disease, ALS. But of course, they're armed with knowledge of the genetics. So they can use iPS lines that carry the risk genes and see how that impacts on the behavior of the cells. So yes, it is a problem, but it is a problem that to some extent you can sort of start to overcome.
Now, in 2014, so Shirasaka and Kurosawa suggested that intravenous transplantation of neuronal stem cells could be used for reconstructing impaired networks in schizophrenia. And in fact, this year in schizophrenia research, so they published a letter to the editor where the authors suggest that they have in fact injected umbilical cord blood cells in patients with schizophrenia and showed increase of brain cortical activity. So are we that close to stem cell therapies, or am I missing something?
Well, this is a very good question. And I have to say, this is something I feel somewhat reasonably strongly about. I published a paper earlier this year reviewing the trials for using stem cells that have taken place in autism. My feeling is it's just much too early. I mean, some of the trials have been well constructed. Those that have been well constructed have largely been negative. So they've largely come up with negative results. The example that you cite is one of the few sort of exceptions. There's a lot of other work going on that I think is really, really quite dubious. Some of them are not really proper trials. They're really pay to participate studies in inverted commas. In other words, you pay $20,000 and you can get stem cells injected into your child. I think some of these are really unethical, to be perfectly honest, and the rationale is really not good yet. The mode of action of the cells isn't very well sorted out. The potency assays for the cells are non-existent. Even the release assays for the cells are poor. I think it's very hit and miss. So I think it's too early.
Okay, so too early. Okay, so that's good to hear. Now, as Solomon commented on a molecular psychiatry review back in 2017, now one of the potential limitations of pluripotent stem cells is epigenetic erasure, meaning that when we take a cell on a journey back in time, we are essentially losing the influence of the environment. And we know how important the environment is for psychiatric disorders. So how can we account for all the added stress that comes with life experiences and trauma and substance misuse that all these can contribute to someone developing a psychiatric disorder? So how can we account for all that on a cell?
Yeah, well, no, that's a very, very good question. One of the things I would say is it's not totally clear the extent to which the erasure of epigenetic signals, epigenetic marks, holds true for those that are associated with disease and with stress. It's certainly true that during normal embryogenesis, epigenetic marks are wiped, and there's good evidence that happens in iPS cells. But it's not entirely clear whether if the cells went through stress, or actually treatment of the patient might be residual in the epigenetic signals within the cell. Not entirely clear if that's wiped. But to answer your question more directly, we can investigate the impact of environmental stresses just as we can genetic stresses. So I talked about the maternal immune activation a few moments ago. as part of that study, we were treating cells with some of the environmental agents that they would have been exposed to in utero. We used cytokines, but you could use toxins or drugs or whatever other factors, might have been environmental factors. So we can actually study that. I think it's a complex story there, and I think more work's got to be done on that. But I think that is something we can do. Possible I've lost the sound. I can't hear you. Is that me or?
Can you hear me?
I can hear you now, yeah.
Yes, we're back. So I'm saying that, Lancaster published a seminal paper in Nature, back in 2013, talking about cerebral organoids, mini brains, if you like. So can we actually grow brains on a fish?
Well, not whole brains, no. But as you say, brain organoids. This is very exciting work, very exciting work. So when we were talking a few moments ago about iPS cells, I said they were pluripotent, meaning they can generate the whole range of different cell types. But in some ways, an even more important property is they have the capacity for histogenesis. Because as I've said, we've had neural stem cells for very, very many years. We've been working on them for many, many years. And they had the potential to make neurons and glia and all the different brain cell types. So you might ask, well, okay, now we have pluripotent cells, they make neural stem cells and they can make all the different cell types. Well, how is that progress? Well, it's progress because the pluripotent cells have the capacity for histogenesis. They don't just make neurons and glia, they build brain tissue. You can actually watch them in a dish starting to make a piece of brain tissue. And as you say, Madeleine Lancaster really was at the forefront of this, although it's now spread broadly. My colleague Deepak Shrivastava is doing this over in the Vole at the present time. That you can actually build actual pieces of tissue and watch the cells migrate, watch them actually go together to start to make a piece of brain. Now, currently, there's a limitation, namely that once they get to a certain size, they kind of need a blood supply. And without it, they start to necrose and, the whole thing falls apart. So that's a real challenge. Nonetheless, up to a certain point, they do start to build brain. So it's very, very exciting, very, very exciting work. They do it on their own. Madeleine Lancaster says this is not so much cell engineering, it's more gardening. You set it up and it does it itself. You just watch it happening. It's very
in August last year, Mortley published a paper in Cell, Stem Cell, reporting the creation of human brain organoids that produce coordinated waves of activity. And this type of brain-wide coordinated electrical activity is, in fact, one of the properties of a conscious brain. And just a few months earlier, a team at Yale University, so they announced that they had at least partially restored life to the brains of pigs that had been killed hours earlier. And in fact, I remember, in one of our experiments, back in Hopkins, we were injecting induced neurons into mice. So this all sounds amazing. But as you would expect, raises endless ethical questions. So what are the boundaries?
Yeah, well, that's a good question. What are the boundaries? I mean, the two examples you give are actually very, very different. I mean, the reperfusion of the pig brain, I think, is really potentially very, very important from an ethical point of view. The iPS stuff is nowhere near far enough along yet to be really concerned about it. But at the same time, I don't think we should be dismissive. I don't think it's an acute concern. And I think your question's the important one. What are the boundaries? And I don't know. I think we're going to have to do some work on that. I don't think consciousness is going to be a useful concept here. There's so little agreement amongst people who even amongst people who work on consciousness, how you would define it. And I think it's very, very unlikely that we're going to grow things where we're going to be able to recognize consciousness in any meaningful sense. Also, people talk about pain. You might be generating something that's feeling pain because of what you're doing to it. But of course, these bits of brain have no sensory input. So again, I'm not too worried about that. So I think you're right. I think this is something we need to think about and how we draw the boundaries. I mean, I think we're a long way away from important boundaries, but how will we recognize when we start to get close to them? And I don't know the answer to that, but I think it is something we need to think about as we move forward.
I mean, it was only this April that you published your new book. It is number 2 in my reading list right now. I need to finish the history of ambient music.
I won't ask what's number one.
You're getting there. You're getting there. Don't worry. So now, your book is called "The Future of Brain Repair: A Realist Guide to Stem Cell Therapy." And although there is a difference between realism and pessimism, it is a fine line between them, right? So tell us a bit about the book, but also why you chose that specific title.
Well, first off, I have to come clean. I didn't pick the title, the publishers. My title was much less interesting than that. They came up with that one. But nonetheless, I like the title. And I think you're quite right. Realist, because I think this causes for optimism, serious optimism. There's also causes for concern. So the optimism is this. So some of the earliest trials, real proper clinical trials, for cell therapies for brain disorders are now starting to deliver. They're coming through phase 3. So we'll find out if they work. Now, some of those very early ones didn't use technology that we now know to be the best, and iPS cells that we'll be discussing is one of those. But at least we'll find out the answer. So they're being properly trialed. Second, the iPS-derived cells. So iPS as a starting material is far superior to the things that I was doing 10, 15 years ago. And some of those therapies are starting to come through as well. And I would say age-related macular degeneration, the replacement of the retinal pigment epithelium, great work being done there. Parkinson's disease, stunning work being done, using iPS to really precisely define a population of dopaminergic neurons that we know will be therapeutic. So those things are really in the offing now. In the next 2-3 years, proper clinical trials will really reveal that they will work, and I'm very optimistic. So that's the upside. The downside is the stuff we've already referred to. So it's what I refer to as feral therapies in the book. And these are therapies that are really on the wild side, people paying vast sums of money to join, trials in inverted commas that aren't really trials. Do you know there are over 300 companies in the United States now offering direct-to-consumer cell therapies? You look online, you go to their clinic, they take quite often cells out of your midriff, fat cells, dissociate them, very poorly characterized, never gone through proper clinical trials, and people are getting them injected. Here's one. Just let me give you one example, right? So some guy, this is a couple of years ago now, turns up in an acute medicine facility in Florida, apparently having a stroke, right? An ischemic event, a serious ischemic event, brain ischemic event. What's it been caused by? He's had cells injected into his scalp. So his fat cells taken out, injected into his scalp to cure his baldness, right? There's lots of examples like that. Right?
Wow.
And it is totally legal within the United States, and it's very, very common. Not here, fortunately. My old colleagues at the MHRA jump on that kind of stuff pretty damn quick, you can be assured. But it's a serious problem. And apart from anything else, this kind of stuff completely discredits the field. And the proponents of that kind of approach are working very, very hard to try and get it broadly accepted, to get the US government to soften their regulations to allow them to get this stuff to market without going through proper clinical trials. I think that would be a disaster if it happened.
And now, Rob, looking at your CV. So, which, to be honest, it's a good introduction to my next question, because looking at your CV, it's really striking that, you served in senior positions, both in academia and the industry. So I will refrain from asking, the cliche, what's better, but instead, so what do they have to learn from each other?
Lots, I would say. If I were to point one thing in each direction, I think what academia adds that industry struggles slightly with is real innovation. So most of the innovation, well, of all the innovations we've been talking about, and lots of other CRISPR-Cas9, single-cell transcriptomics, all of these things, they grow out of academia, and I think long may that continue. And industry recognizes that the academia is much more innovative, probably than industry.
Wow.
The opposite direction, you know what? I think we could learn a little bit about veracity. I'm very concerned about the reproducibility of science. neuroscience particularly, and we clearly fail quite a lot of the time to, we're too often happy just to publish without really being sure that what we're saying is broadly true or broadly applicable. Industry wouldn't put up with that for very long. If you're in a research group in industry, right, and you come up with an assay and you just sling it over the wall into development, and it fails, they waste a year trying to get your assay to work, and they're going to come looking for you. And we don't have an equivalent in academia, right? And I think that's a bit of a problem. So I think if I picked on one thing that each could learn from the other, it's that.
Fantastic. And Prof, last question. So how can we bring neuroscience closer to people like me, like clinicians?
Well, Pospo, do you know what? I think the Institute's already doing this. I think having a cadre of really academically trained psychiatrists, clinicians generally, who really want to engage with psychiatry, with neuroscience and with the latest research is really, really pivotal. And I think we have it at the Institute. I mean, the research we were talking about a moment ago, if it hadn't been for Eva Loth, for Gráinne McAlonan, for Declan Murphy, these clinicians who really, it's not some, yes, okay, we'll help you, but it's all a bit off to one side. These are profoundly involved in the kind of research we've been talking about. And that's the key. And I think in psychiatry, that's getting better all the time. I mean, I remember a time when psychiatry was mostly made up of people who studied medicine but wanted to get away from biology, right? Took one look at a Krebs cycle and run a mile. I don't think that's true. I think we've got, if you were really interested in biology, you went into hematology or immunology or whatever. You didn't go into psychiatry. I don't think that's any longer the case. And I think in the future, I think it's going to be even less the case. And I think it's a great future there for academic psychiatrists. And, we'll both work both ways.
Amazing. BRAINCAST people, Professor Jack Price. Amazing, amazing stuff. I mean, if you cannot get enough of stem cells, just go and buy his book, The Future of Brain Repair, a realist guide to stem cell therapy. But before you do that, though, make sure you update your visas, as next week we're flying to Boston to meet Dr. Vissam Deeb and see how he enhanced people's memories using neurosurgery. No, he didn't take anything out of the brain. It's all about stimulation, deep brain stimulation. Until then, Pospo and BRAINCAST for Mostly Learning, over and out.